Hi everyone. Today I'm going to be explaining how I use a centrifuge in the AMSR lab. This is what it looks like, and it's a machine that rotates mixtures and cultures at a fast enough rate to create different layers in a test tube based off of density. So I use it to separate my Anabina cultures into different layers. So I have one layer that is a solidified pellet of the actual Anabina algae, and then I also have this residue left over called the supernate. And I use both of those in my experiments. So what I do is I put part of that culture into a 50 milliliter tube. I usually use 10 milliliters. And then I label it, I put it in the centrifuge. But in order for the centrifuge to work properly, I have to also have another tube with the same amount of um, materials inside it. So if I have 10 milliliters of Anabina in one tube, I need to have 10 milliliters of water in the other tube to balance it so that the centrifuge doesn't get wobbly, it doesn't break, and it doesn't mess up the results. And so I put those in opposite sides and I label it obviously so I can tell which one is which. And then I close the lid and I can set um, different measurements so I can change how fast it goes, how long it goes, and the temperature. And so I usually use for Anabina 5,000 RPM, which is basically the amount of revolutions the centrifuge does, how many times it rotates, because that's what a centrifuge does is it spins said mixture or culture. And then I also have it at 22 to 24 degrees Celsius, and I do it for about 5 minutes. I changed that since last year because it wasn't working right. But this way, I can end up with a solidified pellet of just straight algae with no water, and I can put that into one of my cultures for my trials with my water bears. And I can put the residue water in another culture with my water bears. So that's how I use a centrifuge. Thank you.